Pyres again. Today I'm going to explain to you how to do this virtual microscope lab. So the procedures are here. This goes from steps 1 all the way to 11. So like I said, always go through and read the procedures first before doing the lab. So I'm going to show you how to actually navigate around a virtual microscope. From the previous slide, you have some familiarity on how to do this, but this time I'm going to show you how to do it using looking through the microscope using different slides. So let's go ahead and follow step 1. It says click on this link to open the tab. So we're going to go ahead. You may already have this open. If not, click on that link and just wait for this link to load. Okay, the next step, if you haven't done so already, you can hit Control minus or Command minus, and this will either zoom out or zoom in. So, sorry, Control plus will zoom in. So what I'm going to do is I'm going to hit Control plus so that I can see this a little clearer. Step 2, in the main menu, click on Explore. So we could see we have down at the bottom, we have the dashboard. We're going to click on Explore. Step 3, open the slide catalog marked with a question mark. So right here is the question mark, and this is called a slide catalog. And this is where we have all the prepared slides. Step 4, select plant slides, then plant cells. So we're going to go ahead and click on plant slides, and then we're going to click on plant cells. So what we're looking at is really not much. It looks as though it's just it's just green. And so this is us as if we're looking through the eyepiece of this microscope. So what we need to do is step 5, adjust the coarse focus first until the picture becomes clear. So down here we have the coarse focus, we have fine focus, and we have light adjustment. We also have 4x, which is the lens that we're looking through right now. If we want to magnify this a little bit more, we can click on 10x, 40x, and 100x. So for this, we want to start with step 5. It says adjust the coarse focus. So we're going to adjust the coarse focus, and we're going to use that until we get to see a little bit more of the image of the prepared slide. As you adjust this, okay, you can actually see this is called a stage of the microscope, and the stage of the microscope will either go up or down. And so if you want to zoom in, you notice how the stage will move up. You want to zoom out, the stage will move down. So we want to get a clear picture of what we're looking at, and so we're going to just adjust it so that it comes into focus. The image comes into focus. Step 6, click on 10x. The image should become clear. Adjust the light focus to brighten the image. So we're going to click on 10x. Okay. We're going to next go to our light adjustment here, and we're going to brighten it. So we are putting a little bit more light so we could see a little better what we're looking at. So if we darken it, it's really hard to see. As we magnify this, we actually lose some light, and so we need to make sure that we adjust the light so that we could see it a little clearer. The next step, we're going to go ahead and click on 40x. So step 7 says click on 40x, adjust only the fine focus until the image becomes clear. Adjust the light focus to brighten the image. So we're going to click on this. Okay, so now we have 40 times magnification of, remember, this is plant cells. So we're going to adjust the light, and we're also going to adjust the fine focus. Now we could see a little bit more clear what we're looking at. And so this, what you're looking at, these are called plant cells. And plant cells are actually, they look like they're lined up like bricks. And this is what Robert Hooke this is how Robert Hooke called them cells, because they look like they were like cells of a monastery. And so down here, okay, this is called the nucleus of the cell. And so this entire portion here, this is a cell. We have another cell here. We have another cell, okay? In the darker parts of this plant cell, this is called a nucleus. And you'll learn more about a nucleus once we get further into this unit. Now, if you want to zoom in further, you can click on 100x. Okay, it's going to tell you to add immersion oil. And what immersion oil does is it just protects the lens so that it doesn't damage the lens when you zoom in closer. So let's go ahead and add the immersion oil. And now we can see even better a plant cell. So this is right here is the nucleus, what you're looking at. Right, so we can adjust the fine focus again, and then we can adjust the light, see a little better. And so the hazy parts, the lines that you see here, okay, this is what separates a plant cell from another plant cell. Okay, so those hazy lines here, this is a cell, here's another cell here, here's another cell, and the darker part inside the cell is called a nucleus. And we can actually start to see other parts of this plant cell. It's called chloroplast. So here are more chloroplasts. The chloroplast is what makes the plant cell green. So what you're going to do is you're going to go ahead and work on slide 7 all the way to 10. And what slide 7 to 10, let's go flip over to that. It's going to tell you here what slide you should be using. Okay, so we did plant slides, plant cells, and then at 40x, meaning 40 times magnification. So let me go ahead and continue here. I didn't get to read step 9 through 10. So step 9, it says draw your observations in your notebook. Remember to try to be as clear as you can when drawing the image, just like Robert Hooke. And then step 10, take a picture of your drawing using the camera on your computer and attach it to the next page. So that's what you're going to do here. Draw the picture of the plant cell at 40 times magnification. Take a picture of it using your camera, and you're going to attach it to this page here. You're going to do the same thing. Let's move on to slide 8. It's going to tell you to go into animal slides. Then you're going to click on spider, and you're going to draw it at 10 times magnification. So let's go back to the virtual microscope. We're going to remove the slide, and it's going to ask you to clean the lens. So we have something called lens paper, and what it does is it wipes the lens so that it's clean so you don't contaminate other things that you're looking at. We're going to click on the question mark. We're going to click on animal slides. This is slide 8 now. And we're going to click on spider. Okay, you're going to do the same procedure. We're going to first use the coarse focus to make the image appear clearer. You can also adjust the fine focus if you need to. Then we're going to move to 10x or 10 times magnification. And you're going to draw at 10 times magnification what you're looking at. You can drag this around so that you could see a little better what the image is that you want to draw. And you can adjust the light if you want to make it clearer. So the only thing that you're drawing here is the 10x magnification. You can explore what it looks like further, 40x and 100x. You can explore that, but in your drawing, in your notebook, you're only drawing 10 times magnification of the spider. In slide 9, okay, slide 9 says bacteria slides. So you're going to go here. It says bacteria slides, gram stain mix at 100 times magnification. So we're going to go back. Once you're done drawing this picture, you're going to click on Remove Slide. Okay, you're going to open this, click on Bacteria Slides. We're going to look at gram stain. Okay, and then you're going to do the same thing that I showed you in the previous, for the previous slides. Okay, so you can go ahead and draw this in your notebook. You're going to start off with the plant slides first. Draw a picture of that in your notebook, take a picture of it and attach it to the slide. Animal slides next. Spider at 10 times magnification, draw in your notebook, attach it to the slide. Bacteria, and then last but not least, the human slide. You're going to click on compact bone at 40 times magnification, draw that and take a picture of it.